Hello and welcome to Science Research Weekly, episode 15, Buzzing Across the Pond. Get ready for jetting black holes, cancer diagnosis with a strip of paper, the mysterious world of freshwater acoustics, frontiers in fungal biotechnology, transcribing, translating audio with buzz, and NSF infrastructure grants. This episode covers materials from April 21st to April 27th. Let's dive into some research articles. I have 7 articles today, from the reaches of space to your backyard pond. The first one is a ring-like accretion structure in M87 connecting its black hole and jet. So researchers were able to observe the jet and shadow of the supermassive black hole at the center of the galaxy M87, helping to understand how jets are formed from black holes. Speaking of cosmic formations, researchers found in a recent article, galaxy interactions are the dominant trigger for local type 2 quasars. They found that quasars, mysterious cosmological objects that can be as bright as billions of stars in the space of a solar system, these quasars were dominant in interacting or colliding galaxies. Switching gears from big to small, we have CRISPR-Cas amplified RNA biomarkers for multiplex and portable cancer diagnosis. So researchers created a microfluidic platform for detecting cancer biomarkers that could be much more affordable and deployable than current methods, while still being accurate enough to detect different tumor types. The modified test uses a paper strip similar to that of something like a rapid COVID test or pregnancy test. Also along the testing procedure, there is the article, Simple Optical Nanomotion Method for Single Bacterium Visibility and Antibiotic Response Testing. So researchers were able to run an antimicrobial susceptibility test, so an AST, using a basic microfluidic analysis chamber, an optical microscope, a camera, and software. It was simple and efficient, 2 vital qualities in the field of rapid AST. Then switching over to a watery world, we have a versatile jellyfish-like robotic platform for effective underwater propulsion and manipulation. So researchers created a quiet and soft-body robot that resembles a jellyfish that could be used for a variety of underwater applications, including exploration, monitoring, and trash cleanup. The ocean's really the limit here. Staying in the water world, but switching to freshwater, there is dial variation in insect-dominated temperate pond soundscapes and guidelines for survey design. So researchers monitored acoustics in freshwater ponds in the UK. They discovered daily patterns of variation and connected acoustic diversity with species diversity. And now, while acoustic monitoring has been used in things like birds and monkeys in the rainforest, or even large mammals in marine water, freshwater acoustics have really remained mysterious. So this sort of helps uncover that mysterious world in a sort of introductory degree. Okay, and then finally, the paper I have is Fungal Biotechnology from Yesterday to Today. Fungi has a great potential in biotechnology, but has remained understudied and underutilized. So moving forward, technologies like long-read DNA sequencing might change that to really open up the field of fungal biotechnology. All right, with that, let's turn to some research tools. So in case you've been under an R rock, R 4.3.0, already tomorrow, released last Friday. And then the analysis factor has a guide on how to pick an R package. Here's the short of it. So start with the CRAN page. Then you can read other sources about the package, like the Journal of Statistical Software or the R Journal, 2 things I mention a lot on this podcast. Yeah. And also you can finish up with checking task views. Speaking of picking R packages, here are new CRAN packages to look over with a discerning eye. There is jmBIG, Joint Longitudinal and Survival Model for Big Data, mlmpower, power analysis and data assimilation for multi-level models, gtranslate, translate between different languages, mldr.resampling, resampling algorithms for multi-label datasets, geessbin, modified generalized estimating equation for binary outcome, and survivalSL, super learner for survival prediction from Censored Data. Okay, there's a couple of interesting projects via GitHub that I wanted to talk about. First is Buzz, with the power to transcribe and translate audio offline on a personal computer. It's powered by OpenAI's Whisper and is available for Mac, Windows, and Linux. The other one is Tabby, a terminal for a more modern age, formerly called Terminus. Tabby is a highly configurable terminal emulator, SSH, and serial client for Windows, Mac OS, and Linux. With tools wrapped up, let's finalize our time here today with research funding. So through the NIH and National Institute on Aging, so the NIA, there is a small research grant program for the next generation of researchers in AD/ADRD research. This is an RO3 clinical trial optional. The program will support meritorious projects to provide needed scientific insight to improve the prevention, diagnosis, treatment, and/or care for individuals with Alzheimer's disease and Alzheimer's disease-related dementias. The NSF has 2 infrastructure grants. The first is Infrastructure Capacity for Biological Research, which supports the implementation of, scaling of, or major improvements to research tools, products, and services that advance contemporary biology in any research area supported by the Directorate for Biological Sciences at NSF. Awards are between $25,000 and $1.2 million. The other one is Infrastructure Innovation for Biological Research, which supports research to design novel or greatly improved research tools and methods that advance contemporary biology for NSF. The awards are between $300,000 and $800,000, so a lower ceiling but a higher floor. And then the Human Frontier Science Program has a long-term fellowship meant for frontier, potentially transformative research in life sciences. Applications for high-risk projects are particularly encouraged. Finally, CAP, the College of American Pathologists, has a global pathology development grant created to improve global health in the science and expansion of pathology and pathology services, up to $10,000 in funding for supporting impactful, sustainable, and accretive projects, international projects to develop, expand, and sustain the delivery of pathology services through education, training, patient care, laboratory quality, or enhanced technology in laboratory diagnostics. Well, busy as a cosmological bee, thanks for joining me for Science Research Weekly. Science on.